Okay.
Hi everyone and hi to Dr. Karyu. So today we are going to present about plant identification with two plant species which are Chiku and Rambutan. So we are from group 3 from Advanced Agricultural Botany. Next. Tak ada orang ke? Okay. Okay, so I will start with Chiku. Chiku, also with its scientific name Manikara zapota. So we can see from the taxonomy tree, it is from Spermatophyta phylum, followed by Angiosperma, Dicotyledone, and family of Sapotaceae, and of course the species name Manikara zapota. Next. So this is the description of Chiku plant. Manikara zapota, also known as sapodilla. Also, we call it as Chiku. So it is an evergreen tree with 5 to 20 meter in cultivation that can reach up to 40 meter in the forest. And this is a tree with round, dense crown. And it is an important fruit tree that planted over Southeast Asia, such as Thailand, Philippines, and of course our country Malaysia. Besides these countries, it is also planted in Asia, Central America, and Florida in US. Next. 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 Okay, so chiku tree can be planted in terrestrial such as primary rainforest. And there are a few types of propagation such as seed germination, cutting layer, air layering, and of course, grafting. And lastly, it has the uses as landscape tree, traditional medicine, foods, furniture, and latex. Next. So these are the characteristics of chiku tree. From the tree leaf, we can see that it is simple, arranged alternately, and the leaf margin is entire, and the leaf shape is elliptic. And for the flower, it is bell-shaped and arranged in cluster, and the flower is white or green color. Lastly, the fruit type is berry. Next. Okay, I will pass this to the second speaker.
Okay, now I will present about the rambutan. This is the taxonomy for the rambutans, and the phylum of the rambutan is Spermatophyta, and the class is Menolipsita, and order for the rambutan is Sapindalis, and rambutan is under the family of Sapindaceae. And the scientific name of the rambutan is Nephilium lepaceum. And rambutan is a shrub-like tree and usually is even green tree, and it usually grows from 9 to 15 meters tall in the wide areas, but it also can grow up to 30 meters. And for the cultivative though it will usually often prune to 4 to 7 meters tall, and it is suitable the climates around the world. And rambutan is also as edible fruits, or also it can be ornamental tree. And the uses of the rambutan is for local medicine use, supplies dyes, oil, and also used as timber. And the characteristics of leaf is simpler pinnate compounds, and the leaf arranged in alternate, while the leaflet is arranged in opposites. Usually, one leaf will have 1 to 6 pairs of leaflets, and the margin is strongly curved and nearly straight and parallel, and the shape is ovate to ovate, and the flower of the rambutan is greenish white, small and large bunches, and the fruit is round with leathery skin with soft spines, and the fruit is white juicy flesh and can cross and overseed. Next, I will pass to the next presenter, Shukran.
Okay, hi. My part, I will present about the plant histological procedure. This procedure contains 9 steps. The first step is fixations, where the aim of the objective of this procedure is to stop all the cell activity. Okay, then the second procedure is vacuum. The objective of this step is to suck out all the air from the specimen. Third, the dehydration process, where to suck out all the water from the cell. Fourth, infiltration, to suck out ethanol from specimen tissue. This is because from the dehydration process, we use to dry the plant cell, we use ethanol. So to remove all the ethanol, we must use the infiltration process. Then the fifth, embedding to arrange the specimen into a section such as stem, root, and etc. Okay, next is sectioning, where we cut the plant into each section with careful procedure. Then the next process is mounting. This is to stick the specimen section to carotene slide using wax. The next is to color the specimen, where we use xylene and ethanol to make the specimen color. So then the lastly is finishing, where we mount the specimen perfectly onto the glass slide. So that's all from me. I will pass from to the next presenter.
Okay, thank you. So now, as we know, Chiku and Rambutan are both cotyledon plants, so their microscopic results supposed to be very similar. So let's take a look on the Pto cross-section. So in the Pto, we have a concave and convex shape. We have two more prominent regions on the abaxial surface, as you can see on the slide. And in the epidermis was composed of a single layer of the cell and covered with a very thick cuticle, which labeled as a CT for the cuticle and the EP for the epidermis. And for the parenchyma, as I labeled for CO on the left side, was composed of 3 to 6 layers of the cell and also present in the ribs of the abaxial surface. And in the most internal layer was formed of parenchyma labeled as the PA. And in the cortical region of the Pto was present of the isolated scarate and a group of fiber as well as the latissifer and the lignified cell. And for the latissifer are labeled as the LA, and the lignified cell is LIC. And next in the medullary region, the latissifer and the nucleus of the phloem also been found, accompanied by the lignified cell. Okay, so next slide. So now is the leaf lamina or the leaf blade. So in the leaf lamina cross section, the epidermis coated with a thick cuticle as well, and the midrib have a concave to convex shape too. And the parenchyma was arranged in 3 to 6 layers of the cell, and being more developed in the abaxial region. Next, for the vascular system was disposed in the same way as well that observed in the Pto too. And there was a presence of the latissifer and the lignified cell. And one more thing is the prismatic crystal in the cotyledon cortical region, which I label as the as you can see in the slide. And in addition, the latissifer and the nucleus of the phloem also accompanied by the lignified cell. And in the leaf lamina, the stone cell will also be identified just next to the vascular system. So that's all from me. Thank you. And now I'll pass my turn to Adit. Thank you.
All right. Hello. Assalamualaikum. So I will continue. I will continue from your part. My part is stem and roots. So firstly, as we can see in the stem cross section, the epidermis covered with thick cuticle and lenticel in primary growth. We can see the primary growth in these figures 1A and B. So that is the first primary growth. So in secondary growth, in figure C, we can see that the peridium, development of peridium was observed in figure C. In the cortical region of the stem, both primary and secondary lactifiers, lignified cell, and prismatic crystal in figure A and C were found. So in the pith of the stem, both primary and secondary growth, only lactifiers and starch grains occurs. That is on figure E and also figure F. So that is the what we call it, the abbreviation for the term CO, CT, CO for collenchyma, CT for cuticle, and more of it. All right, next. All right, next I will explain. This is about the cross section of the dicot roots for both rambutan and chiku. It had a single layered and composed of thin wall epidermis cell, which we can see at the upper of the root cell. Then we go down a little bit, we can see the cortex. The cortex is the last layer. Last layer of the cortex is endodermis. The endodermis composed of one layer of barrel-shaped cells which are closely arranged without having intercellular space. All right, next to endodermis, there is a single pericycle, as we can see, made of thin wall parenchyma cell. The pericycle is the seat of the origin of lateral roots. And the last one is conjunctive tissue. It's a thin wall parenchymatous cell lying between xylem and phloem groups. At the center is a small parenchymatous pith. You can see there is a pith in the diagram. So it may even absent in some of the dicotyledonous roots. So I guess that's all from me, and that's all from us. Thank you so much.